So our goal here is to inoculate colonies from some of these metagenomic libraries onto membrane filters. And each of these filters can hold 10,000 colonies. So what Saltram's doing right now is laying this nylon filter onto a block and kind of smoothing it out. It's in a little bit of liquid. It's just the growth media, luria broth. And what he's then going to do is transfer this moistened membrane over to the robot that is going to automatically inoculate this membrane with 10,000 colonies. And robotics are essential for this kind of procedure because no human being really has the patience or accuracy to spot 10,000 colonies onto one of these membranes. Robots are one of the things that has actually advanced genome science by allowing very repetitious and kind of small spatial scale operations to be done with high accuracy. Right now, Saltram's setting up the robot to feed these 384 well plates. Each well in those 384 well plates holds an individual colony from the metagenomic library. Right now, the robot's sterilizing the inoculating needles that it's going to use to spot on the membrane. So it goes through several different solutions, one of which is 70% ethanol, kind of sterilize it, and then those inoculating needles are going to get dipped into a 384 well plate that has all the colonies. Now it's being sterilized and dried by light.